Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing DBT JRF entrance exam question paper conducted in the year 2019. This is the part 2 of the question paper. Part 1 I have already done. Coming to the first question. A chemist synthesized a new chemical X which is highly mutagenic. He also tested the capacity of mutation induced by X to be reversed by other known mutagens and obtained the following results. So, which one of the following conclusions is appropriate? So, if you see nitrous acid, it is reversed by nitrous acid. So, nitrous acid produces exclusively transition mutations, and because both CG-TA and TA-CG transitions are produced, these mutations can be reversed with nitrous acid. So it shows that X causes a transition mutation. That is why it got reversed with nitrous acid. Whereas hydroxylamine add hydroxyl group to cysteine, converting it into hydroxylaminocysteine. This conversion increases the frequency of a rare tautomer that pairs with adenine instead of guanine and leads to CG-TA transition. So hydroxylamine, it causes CG to TA transition. Because hydroxylamine acts only on a cysteine, it will not generate the TA-CG transitions. Thus, hydroxylamine will not reverse the mutation that it produces, whereas nitrous acid will reverse. Then, what would be the best assay to detect and quantify a small and low abundant peptide in a biological sample? So it is radioimmunoassay. It is an in vitro assay that measures the presence of an antigen with very high sensitivity. Basically, any biological substance for which a specific antibody exists can be measured even in minute concentrations. So the answer is option C, radioimmunoassay. A mutation in the coding region of a mammalian gene leads to a loss of single amino acid at the N-terminus of the nascent polypeptide. This is possible when? So there has been a mutation in the coding region, and even then a nascent polypeptide has been synthesized. We know that the amino acid at the N-terminus is methionine. Now if methionine itself is not present, then translation will not take place. But translation has taken place. Now if translation has taken place, then there should be two methionine coding regions in the gene next to each other. So when first methionine is removed, the second methionine will function properly. So the answer is the first two codons code for methionine. Answer is option C. Then next one. You have two tubes containing bacteriophage labeled with radioactive phosphorus, tube A, and radioactive sulfur, tube B, that are devoid of bacteria. You use these bacteriophages to infect separate E. coli cultures. After infection, you separate bacteria from the virus and check them for radioactivity. You will find. So radioactive phosphorus will label the DNA of the phage, and sulfur, radioactive sulfur, it will label the protein code. So upon infection to E. coli, radioactive phosphorus, which is labeled to DNA, will enter the cell, whereas the protein code will be left outside. So you will see radioactivity in bacteria infected with virus from tube A. So answer is radioactivity in bacteria with virus infected from tube A. Answer is option C. Next one. A scientist performs a series of experiments to determine the recombination frequencies between the following genes. He acquires the following data. Which one of the following represent the correct order of the genes? So I have done it. You can just go through that. The answer is SPQR. Then next one. Color blindness in humans follows sex-linked recessive mode of inheritance. If a couple with normal color vision has a color-blind son, what will be the genotypes of the parent? So color blindness is sex-linked recessive, so normal female can have carrier genotype. So capital B is for color blindness and small b is for normal vision. So the female is heterozygote and a carrier, whereas the male is normal visioned. So these are the gametes they produce, and upon crossing you will get a color blind son, that is this one. So here in the option they have given capital, in option B they have given X capital B, showing that the male is also infected. That is a printing mistake. It must be small b. So the answer will become option B. Then next one. If the allele A is incompletely dominant over allele small a, what is expected in a progeny of two heterozygous parents? So these are the heterozygous parents. You will get genotypic and phenotypic ratio as 1:2:1. So same phenotypic and genotypic ratios. Answer is option A. Then an experiment involves formation of RNA-DNA hybrid. Which one of the following enzymes could be utilized to degrade only the RNA strand from the RNA-DNA hybrid? That is RNase H. Answer is option C. If the intracellular pH of a cell becomes basic, which one of the following will help reduce the pH? Now HCO3 minus is basic, and so remove it and import Cl minus ions which are acidic. So import Cl minus and export HCO3 minus. Answer is option B. Proteins are an excellent buffers because the wide range of pK values of side chains found within the proteins. Answer is option A. Which one of the following statement is incorrect in relation to reverse phase chromatography? So the answer here is the solutes elute with increasing order of molecular weight. That is incorrect in relation to reverse phase chromatography. Reverse phase chromatography doesn't separate molecules on the basis of their molecular weights. The DNA gel picture shown below depicts the PCR banding pattern of two markers M1 and M2. The linkage distance between the two markers from the test cross population is. Now this is marker 1, marker 2. P1 is the parent, P2 is the second parent, F1 is the progeny. Now if you see P1, P2 and F1, the same bands have been obtained in M2 also. But if you see the 10th, 11th band, there is a change between the two markers M1 and M2. So that represent the recombinants. So 1 to 10 and 13 to 25, they are parental combinations. Only 11 and 12 are recombinant. So recombination, what they have asked, the linkage distance between the two markers from a test cross population is, so 2 by 25 into 100, that is 8 centimorgan. Answer is option C. For metabolic engineering in plants having improved tolerance to osmotic stress, mannitol is overproduced because osmotic stress is sudden change in the solute concentration around a cell, causing a rapid change in the movement of water across its cell membrane. Osmotic stress leads to efflux or influx of water from or into the cell. It is hyperosmotic stress, it causes shrinking, and if it is hypoosmotic stress, it causes swelling. Now mannitol, it is an osmolyte. If mannitol concentration increases, we are increasing the concentration of solute. Water will enter the cell. So answer here is lowers the water potential in the plant cell. So water will enter the cell. Now for engineering virus resistance in plants, which one of the following viral component is commonly targeted? It is the coat protein. Answer is option A. The next one, the Bt protein employed for raising insect resistant plant is not toxic to humans because, so it is inactive under acidic pH because our stomach digests the protein, and due to this the toxin is not toxic to humans, whereas the insect gut, it is around pH 10. So the answer here is it is inactive under acidic pH. Then an isoenzyme may be a monomer or a dimer or a multimer with the identical or distinct subunits. Following picture depicts a native page profile of the isoenzyme from a diploid plant. So P1 is parent 1, P2 is parent 2 and F1 is the progeny. This isomer is. So if you see F1, you can see the parent 1 band and parent 2 band, and in between them there is a third band, that is that of the F1 band, and it is a dimer. It is a combined band of P1 and P2 because since it is a native page and no denaturation of the sample is done. So this is a homodimer. Answer is option B. Then next one. Which one of the following sets is an example of specific sequence-based PCR analysis in marker-assisted selection? So DAF, it is DNA amplification fingerprinting. It is a PCR type amplification of genomic sequences using short, single and arbitrarily chosen primers. Then RAPD, it is random amplified polymorphic DNA, is PCR technique which also uses arbitrary primers which bind to non-specific sites on the DNA and amplify the DNA. Then SCAR is sequence characterized amplified region are PCR based markers that represent single genetically defined loci that are identified by PCR amplification of a genomic DNA with pair of specific oligonucleotide primers. And simple sequence repeat or SSR is anchored PCR amplification, is a genetic typing technique based on primers anchored at the 5' or 3' ends of microsatellites at high primer annealing temperatures. And COS, that is conserved ortholog set, are highly conserved with low copy number region. So they have asked out of the following, which one of the following set is an example of specific sequence based PCR analysis? So specific sequence based analysis is taking place in SCAR, SSR and COS. The other two, DAF and RAPD, are using arbitrary primers. So answer will be option B. Then next one, opaque 2 gene in maize and WX gene in rice affects the dash and dash content respectively. So the opaque 2 gene in maize encodes a transcriptional activator that controls expression of various genes during kernel development, particularly some of the most abundant endosperm storage protein genes. And in rice grains, the waxy gene, WX gene, is responsible for the synthesis of amylose. So the answer here is option A, protein quality and amylose. Then next one. Which one of the following is associated with RNA-induced gene silencing in plants? So it is DNA methylation. Answer is option A. Then next one. The oxidative photosynthetic carbon cycle salvages. The oxidative photosynthetic carbon cycle or the C2 cycle or photorespiration, and they have asked its cycle salvages C2 carbon. Answer is option D. Then next one. The term co-suppression was coined by Richard Jorgensen to explain. So in co-suppression, when a gene is introduced by transformation into a cell, neither the resident nor the transgene copy of the same gene is expressed. Now regarding co-suppression, they have not asked the same question, but something related to co-suppression has always been asked in DBT exams. Most of the papers, 2 or 3 papers I have seen co-suppression being asked. Similarly, regarding BT toxins and BT cotton also, they have asked questions regarding coming from BT cotton aspect. Then in co-suppression, when a gene is introduced by transformation into a cell, neither the resident nor the transgene copy of the same gene is expressed. So when you introduce a new gene which is also present in for endogenous which is similar or which is same as the endogenous gene, then neither the endogenous gene nor the introduced gene will express. That is called as co-suppression. So Richard Jorgensen and his colleagues were attempting to create petunias with more deeply colored flowers by overexpressing the gene coding for chalcone synthase, an enzyme that plays a key role in producing the pigment anthocyanin. But they did not get what they wanted. So what they did was they took the chalcone synthase gene and introduced it into the petunia for getting more deeply colored flowers. So chalcone synthase gene is already present in the petunia. Now they introduced chalcone synthase gene for overexpressing that particular gene. So they thought that by overexpressing they will get more deeply colored flowers. But instead of producing deeper purple flowers, many of the transgenic plants had white pigmented free flowers and others had flowers with white patterns on a purple background. So the answer here is option A, patchy flower color in petunia. Answer is option A. Then which one of the following plants exhibits both C3 and C4 pathways? So it is Ziamase. Answer is option A. Dichogamy refers to phenomenon in which anther, dehiscence and stigma respectively are. So dichogamy is a temporal flowering system in which male and female reproductive organs mature at different times within a single flower or across multiple flowers. So the answer here is temporarily separated. Answer is option A. Then light compensation point is the irradiance at which. So light compensation point is the irradiance at which photosynthesis equals respiration. Now why is the answer here is net carbon dioxide exchange is 0. Now why is it so? Leaves release carbon dioxide by photorespiration and cellular respiration, but carbon dioxide is also converted into carbohydrate by photosynthesis. So assimilation is therefore the difference in the rate of these processes. So it is 0. Answer is option C. Then fusicosin promotes cotyledon growth by promoting. Now fusicosin, it is a fungal toxin and fusicosin induces cells to excrete H+ and that results in acidification of cell wall which lead to cell wall loosening and thus cell elongation and thereby growth. So the answer here is option D, that is cell wall acidification. Then next one. Plants take up water from the soil predominantly by apoplastic and symplastic mode of transport. Which one of the following statement is true? Now apoplast and symplast are the two routes by which the water travels from root hair cells to the xylem of the root. In the apoplastic route, water moves through the cell walls and the intercellular spaces of the root cortex. In symplastic route, water moves through the protoplast or cytoplasm of the root cortex. So they have given answer as option D, that is both apoplastic and symplastic transport are ATP independent. Answer is option D. Then which one of the following is molybdenum iron containing protein? So the enzyme nitrate reductase are molybdenum enzymes that reduce nitrate to nitrite. Nitrate reductase. Now we will see for nitrogenase. The enzyme complex nitrogenase consists of two proteins. A molybdenum iron protein, protein 1 that binds nitrogen, and a smaller iron protein that acts as an electron donor to protein 1. So the one which contains molybdenum iron containing protein is nitrogenase. Answer is option B. Then next one. TIR1 an auxin receptor is a. It is a F box protein. Answer is option A. Now phytochromes A and B maximally absorb light at wavelength range. At wavelength range 600 to 750 nanometers. Then phytochrome A is sensitive for far red light, that is 720 nanometers, and phytochrome B is sensitive to red light, that is 680 nanometers. So answer is option D. Then next one. A researcher wants to ectopically express protein X exclusively in the seeds of Arabidopsis. Which one of the following promoters is most suitable for this purpose? So ectopic expression is occurrence of gene expression in a tissue in which it is normally not expressed. Now protein X is not normally expressed in seed. Now the researcher want to express X in seeds only. So napin is a seed specific promoter. Answer is option C. Then given below are the names of different phytohormones in the left column. Match them with their corresponding precursor molecules in the right column. So auxin. Auxin is L-tryptophan. That is a precursor for auxin. Jasmonic acid, alpha-linolenic acid, ethylene, methionine, brasinolide. It is campestrol. So the answer here is option B, that is A2B3C1D4. Then which one of the following is a sulfur containing secondary metabolite in mustard plants derived from glucose and an amino acid? So it is glucosinolase. Answer is option A. Now, the disarmed Ti plasmid of agrobacterium tumefaciens does not result in crown gall phenotype since it does not possess IPT and IAA genes. These are required for auxin synthesis. Now, disarmed Ti plasmid does not have IPT and IAA genes, so it does not result in a crown gall. Answer is option A. Then next one. A plant that survives a local pathogen infection often develops increased resistance to a subsequent attack by a mechanism called. So it is called systemic acquired resistance. Answer is option A. In genetically modified Dhara mustard hybrid 11, male sterility is conferred by dash while dash restores fertility. So male sterility is conferred by Barnase and Burstar restores the fertility. Now a question regarding Barnase and Burstar have been asked in one of the previous years. I don't remember the year. You can refer my channel for previous year questions. Now glycosylation of a monoclonal antibody does not affect fluorescence spectrum of antibody. Answer is option D. Now match the common antibody origin with appropriate generic name branch of the antibody. So mouse it is binatumomab. Chimeric is rituxan. Humanized is Herceptin and human is pantimumab. So the answer here is option C, that is A1, B4, C2, D3. Then next one. A recombinant therapeutic protein is intracellularly produced in soluble form using E. coli. Which one of the following sequences of chromatographic separation method is preferable for obtaining clinical grade protein? Now they have told it is soluble protein, so it is hydrophilic, so we can go for hydrophobic interaction chromatography. And hydrophobic interaction chromatography is often used in combination with techniques such as ion exchange or gel filtration chromatography. So the answer here is option A. Hydrophobic interaction followed by ion exchange. The next one. What property is involved in the separation of a mixture of enolates using gas chromatography? So it is partitioning. Answer is option A. Then next one. Which organization in India approves and gives regulatory clearance of biologicals? So it is the Central Drug Standard Control Organization, CDSCO. Answer is option A. Then next one. 1 microgram of pure enzyme, molecular weight 92,000, catalyzed a reaction at a rate of 0.5 micromoles per minute under optimum condition. The specific activity of the enzyme, that is in micromoles per minute divided by per milligram protein. So Vmax they have given as 0.5 micromoles per minute. Specific activity is total activity by amount of protein. So 0.5 divided by 1 microgram or 0.5 divided by 10 raised to minus 3 milligram, because it is in amount of protein is in milligrams unit. So that will give 0.5 into 10 raised to 3 or 500 units. Answer is option C. Then next one. The ion transport that will be most affected following mutation in the cystic fibrosis transmembrane conductance regulator gene is chloride. Answer is option C. The organism in which the luciferase gene is termed as Lux gene is. So it is bacteria. Answer is option C. The first humanized monoclonal antibody approved by the US FDA for targeted treatment of breast cancer is. It is trastuzumab. Answer is option A. Then which one of the following statement is incorrect with regard to DNA vaccines? No risk of infection. Proteins produced are likely to be correctly post-translationally modified. It can persist for an extended time period in the cell. Introduced DNA stimulates protective immune response. That is not correct. So the DNA vaccine uses a gene from a virus or bacteria to stimulate the immune system. When the DNA vaccine is administered to a patient, the machinery in their cells makes a viral or bacterial protein which their immune system recognizes as being foreign to the body. So introduced DNA doesn't stimulate a protective immune response. It has been the machinery in the human cells or to whom the DNA vaccine has been administered makes a viral or bacterial protein which the immune system recognizes as being foreign to the body. So the answer here which is incorrect is option D. Then antibiotic bleomycin is secreted by, it is secreted by Streptomyces species. Answer is option C. Then zinc deficiency among children primarily results in the atrophy of thymus. Answer is option A. Antigen activated B cells differentiate into antibody producing plasma cells in lymphoid follicles. Answer is option A. Allergenicity of a protein refers to its capacity to activate mast cells. Now IgE produced in response to the allergen binds to the high affinity receptor for IgE on mast cell, basophil and activated eosinophil. So they have given mast cells in the option, that is the correct answer. Now secondary immune response to a hapten depends on. Now what is secondary immune response? When B cells and T cells replicate during the primary immune response, they produce effector cells and long-lived memory cells. Memory B and T cells are antigen specific and encountering the antigen again can mount a more rapid and effective immune response, and this is known as secondary immune response. So they have asked for haptens. Secondary immune response to a hapten depend on. So haptens are small molecules that stimulate the production of antibody molecules only when conjugated to a larger molecule called a carrier molecule. The hapten carrier complex stimulates the production of antibodies which the unbound hapten cannot do and become capable of eliciting an immune response. So both hapten and carrier used in primary immunization have to be used for secondary immune response in hapten. Then next one. IgG class specific antigenic determinants are known as. So they are class specific. So structural differences in the constant region of the heavy chain or light chain determine immunoglobulin class, subclass, types and subtypes within a species. These constant region determinants are called isotypic determinants or isotypes. So the answer here is option B. Then isotypic determinant of IgG molecules are located in the, I just now said, in the constant regions of heavy chain and light chain, HC and LC. Then a chimeric organism is generated when? So a cell of different genetic constitution appear in the same organism. Answer is option A. Then metaplasia represent. Metaplasia is the replacement of one differentiated somatic cell type with another differentiated somatic cell type in the same tissue. So the answer here is transformation of one differentiated cell type to another. Answer is option B. Then the taxonomic method for identifying an organism to its species is, it is DNA barcoding. Answer is option B. Then in Drosophila, the growth of legs on the head instead of antennate during development is an example of. So after the segmentation genes have been established, the number and orientation of segment homeotic genes become active and determine the identity of individual segments. So eye normally arise only on the head segment, whereas legs develop only from the thoracic segments. Mutation in the homeotic genes cause body parts to appear in the wrong segment. So the answer is homeotic transformation. Answer in Drosophila, the growth of legs on the head instead of antennae during development is an example of homeotic transformation. Then amyloblasts are differentiated cells. So amyloblasts are one of the group of cells originating from the ectoderm from which the dental enamel is developed. So amyloblasts are differentiated cells that secrete enamel. Answer is option D. Then which one of the following contributes to the development of the reproductive tract in male fetus? So anti-Mullerian hormone is made in the reproductive tissue of both male and female. Now anti-Mullerian hormone, it plays an important role in the development of sex organs in an inborn baby. During the first week of pregnancy, a baby will start developing reproductive organs. The baby will already have the genes to become either a male XY genes or a female, that is XX genes. If the baby has XY genes, then high level of AMP or anti-Mullerian hormone is made along with other male hormones. This prevents the development of female organs and promote the formation of male organs. If the unborn baby has female XX genes, small amount of anti-Mullerian hormones are made. This allows for the development of female reproductive organs. So which of the following contribute to the development of reproductive tract in male fetus is anti-Mullerian hormone? Answer is option C. The neurotransmitter at the neuromuscular junction is acetylcholine. Answer is option C. Viral vector that is ideal for expressing therapeutic gene in non-dividing cell is, so it is lentiviral vector. They are derived from human immunodeficiency virus that can transduce non-dividing cells. Answer is option A. Then which one of the following amino acids can be used as a diuretic because of its importance in metabolism of ammonia? Answer is asparagine. It helps to break down toxic ammonia within cells. Then next one. Paralytic shellfish poisoning is a foodborne illness that typically develops after consumption of shellfish contaminated chiefly with the heat stable and acidic stable toxin called as saxitoxin. This is also a repeated question. It has been asked some 2-3 times. for DBT exam. You can refer my previous papers. I have uploaded them. Then, the antifreeze molecule that prevent intracellular ice formation in marine organisms are generally So, antifreeze glycoproteins inhibit ice growth via adsorption inhibition mechanism. First, there is irreversible binding of antifreeze glycoprotein to embryonic ice crystal and the inhibition of further growth. So, antifreeze molecules that prevent intracellular ice formation in marine organism are generally glycoprotein. Then which one of the following transgene expressed in transgenic fish by an appropriate inducible promoter may be used for detecting environmental toxicants? So it is green fluorescent protein. Answer is option B. Ballast water may be carried on board by ships to maintain stability and improve maneuverability during transit. Introduction of which one of the following is regarded as the major threat in release of untreated ballast water? So it is invasive marine species. Answer is option C. Then remote sensing of ocean atmospheric parameters carried out in microwave channel is based on the phenomenon of option A, emission. Answer is option A. Then, a representative sequence profile of a given nucleotide binding domain is to be used to mine related sequences from TREMBL. The database to be used to extract the query corresponding to this fold is PFAM. Answer is option B. The figure represents dot plot comparing two genomes X and Y. The portion marked N is an inversion. So if you see a dot plot like this, in this especially, it is called an inversion. Then two types of pairwise sequence alignment of the same hypothetical protein sequence fragments are illustrated in the figure below. Vertical bars between the sequence indicate presence of identical amino acids. The star symbol in figure Y indicate residues not included in the alignment. Which one of the following is correct? So pairwise sequence alignment is the procedure of comparing two sequences by searching for a series of individual characters or patterns that are in the same order in the sequences. So global sequence alignment, an attempt is made to align the entire sequence, end-to-end alignment. And global alignment contain letters from both query and target sequences. If the two sequences are approximately of the same length and are quite similar, they are suitable for global alignment. As in the case of X, they are end-to-end alignment. So that is global alignment. Now local alignment, it finds local region with highest level of similarity between two sequences. That is in the case of Y. It has not included some parts of the alignment. Only those with highest level of similarity between the two sequences is chosen. So a local alignment aligns a substring of the query sequence to a substring of the target sequence. So answer here is option A. Figure X and Y are examples of global and local sequence alignment respectively. Answer is option A. Then match the items in group 1 with group 2 with reference to a database search for identifying homologs of human hemoglobin. So sensitivity, measure of how many correct hits are found. 100% sensitivity, measure indicating that all correct hits are found. Specificity, measure of how many hits found are correct. And 100% specificity, measure indicating that all hits found are correct. So answer is P1, Q3, R2, S4. Option A is the answer. So coming to the next question. Genes are proteins that display the same activity but have different origins and are product of convergent evolution are called as analogs. Answer is option A. Match the type of BLAST program given in group 1 to the particular type of sequence search task described in group 2. So the answer here is option A, that is 2p3q. T-BLAST X is for a nucleotide sequence is to be used as a query to search for similar proteins against a nucleotide database. And 3q BLAST X, a nucleotide sequence is to be used as a query to search against a protein database. Then, in multiple sequence alignment of homologous protein sequences, a region of multiple insertion and deletion indicates the presence of surface random coil region. Answer is option C. So, in multiple sequence alignment, we try to align 3 or more related sequences so as to achieve maximal matching between them. Beta strands and alpha helices are formed in the core of the protein and they are not easily mutated in different organisms. But these residues found on the surface of protein amino acids are easily mutated and they exist as random coils. So a region of multiple insertion and deletion indicate the presence of surface random coils in multiple sequence alignment. Then next one. Two charged molecules are at a distance r from each other. Which one of the following is correct regarding the electrostatic interaction between them? So answer is the interaction energy is proportional to r raised to minus 1 and the force is proportional to r raised to minus 2. Answer is option A. Then next one. A novel protein from a deep sea archaebacterium was identified and sequenced. The sequence is expected to be widely divergent from known sequences. Which scoring matrix will produce the most appropriate alignment in search for homologs in the NCBI database? The one which allows more number of substitution. Now Blossom is strict for substitution and even PAM1. So PAM 250 is not so strict, so you can go for PAM 250. Answer is option D. The measured values of main chain torsion angles of a residue in a polypeptide has values psi and phi as positive values. What type of secondary structure is most likely to be present in? So it is both are positive values and that will come for left-handed alpha helix in the Ramachandran plot. Answer is option A. Then next one. Which one of the following is not an assumption of an evolutionary model of the PAM matrix? So probability of a mutation is dependent on the position of the mutation. So answer is option B. Then next one. Which one of the following is a valid assumption regarding molecular clock hypothesis in evolution? So molecular clock hypothesis states that DNA and protein sequences evolve at a rate that is relatively constant over time and among different organisms. So which one of the following is valid assumption regarding molecular clock hypothesis is for a given protein sequence, mutations accumulate at a constant rate in all lineages. Answer is option A. Then last one. Which of the following is not true of protein folds? Every protein need not fold to function. Okay, so proteins assume a limited number of folds. Proteins with the same fold may perform different functions. Proteins with different fold carry out the same function. A stable fold is a prerequisite for the function of all proteins. I told you earlier, every protein need not fold to function. So it is not a prerequisite. Answer is option D. So that's it. Thank you for watching my channel. Please do like, share and subscribe. I have also uploaded previous year's DBT papers. You can go through them also in my channel. So thank you.